Hello and welcome to this Nature.com custom webcast titled Building Better Therapeutics with Metabolomics. My name is Joshan Karpan and I will be your moderator. Today's webcast will be given by Dr. Dave Morris and Dr. Kirk Beebe and is sponsored by Metabolon. Metabolon is a pioneer and leader in metabolomics, an integrative, powerful phenotyping technology for assessing health. Their proprietary platforms and informatics systems are delivering biomarker discoveries, innovative diagnostic tests, breakthroughs in precision medicine, and robust partnerships in genomics-based health initiatives. We'll begin today's webcast with presentations from each speaker and end with a Q&A session where we will address the questions that have been submitted by you. To ask a question, just press the Ask a Question button at any point during the webcast and we will answer them today. It is now my pleasure to introduce today's first speaker, Dr. Dave Morris. Dave is a pharmaceutical scientist with over 15 years of experience leading multidisciplinary international line and matrix research teams spanning target identification to clinical candidate selection. Over to Dave.
Thank you for the introductions. Kirk and I are cognizant of the fact that the audience for today's presentation will have different levels of exposure and experience with metabolomics. With that in mind, the webinar will feature a short primer on the science and utility of metabolomics in the context of pharma and biopharma research and development. We will then exemplify using case histories how the platform has been and is being used by groups in academia and industry. Specifically, we'll focus on a trend of greater use of the platform in clinical settings and the reasons we believe are behind this trend. We'll wrap up the seminar with some key points and tell you about an exciting opportunity to access the Metabolomics platform at Metabolon. So what is metabolomics and what are metabolites? In this slide, examples of what we mean when we say metabolites are listed on the right-hand side of the slide. The slide also depicts the relationship between the genome, transcriptome, proteome, and metabolome. The metabolome is a collection of small molecules that are at the confluence of the active biological networks in the body. So to summarize, metabolomics is the technology for comprehensively profiling the metabolites that circulate in the body and the interpretation of those metabolite profiles in the context of the body's functional state. Now, to be clear, it is not only genetics exerting an influence here. Metabolomics directly assesses the phenotype, and that phenotype is responsive to changes induced by genetics, external factors, and treatments. So metabolomics is an ideal way to examine how the body processes, integrates, and aggregates these influences to yield a phenotype. Following on from this, metabolomics is a very useful platform for tracking, monitoring, and ultimately understanding the connection between these influences and the phenotype. As you will see from this slide, most of the studies undertaken to date using the metabolomics platform at Metabolon have focused on the discovery and preclinical space. The platform has been applied across a broad range of scenarios. In many, if not all these situations, metabolite measurement and analysis has helped build confidence through greater understanding of the biological pathways responsible for the phenotype. Invariably, metabolomics data helps assess risk and provides reasons to believe in moving in a certain direction as opposed to being unsure and having no reason not to progress. And just to provide a couple of examples, this slide illustrates how metabolomic analysis has been used to identify a new target for sickle cell disease. In this particular case, it was a G-protein coupled receptor, and the paper has been cited in our appendix. It was a Nature Medicine publication. And also to execute a deep biology dive to understand the consequences of PD-1 and CTLA-4 ligation to T cells. Again, references for both these papers will be available in the appendix. As I mentioned earlier, metabolomics has been and continues to be a valuable preclinical platform technology. Interestingly, over the past several years, the number of metabolomic studies aimed at addressing questions in the translational and clinical areas has doubled. We believe this is a reflection of the progressive approach and forward thinking by specific pharma companies and their partners who are all working to build confidence and gain support for moving a program to its next development milestone. Here are some of the reasons for this trend. Briefly, the first point relates to the fact that the current model in the pharma business comes with a highly undesirable rate of attrition. So there is substantial pressure to find new ways to deliver more products. The second reason speaks to the issue that genomic-led insights need to be brought to life with a functional interpretation. Thirdly, metabolomics produces a readout that reflects the functional status of the body. And finally, there have been significant advances in metabolomics technology leading to greater capacity, depth of coverage, and precision of measurements. Kirk will be going into more detail on each of these topics in the next series of slides.
Thanks, Dave. Our second speaker today is Dr. Kirk Beebe. Kirk has been a scientist for over 2 decades with an overarching interest in translating technological and method innovations to addressing fundamental biological questions. He is currently Director of Application Science at Metabolon. Over to Kirk.
So I'm going to elaborate on those points that Dave just mentioned on the next few slides, starting with that first driver of drug attrition pressures. And, because it's well appreciated that this high attrition rate is producing an immense amount of pressure to increase clinical success rates. It's really pushing a lot of individuals to looking for tools and technologies that can ultimately help build confidence and discharge risk, increasing clinical success rate, essentially trying to define a clearer path to the clinic. The second point is really a critical one, and it's what we describe as genomic-led insights, which are really the byproduct of the last 15 years of genome sequencing and all the data we've gotten. And what it's told us is that the majority of the most prevalent diseases, cardiovascular disease, autoimmune disease, Alzheimer's disease, are really a complex combination of genetic influences and environmental influences. And if that wasn't complex enough, it's magnified by the fact that when you consider that most of the genetic influence is distributed across the genome, only a minority of cases are monogenic, it really expands the complexity of the picture. You add to that the fact that on the diet, lifestyle, environment side, there's this whole second arena of the so-called second genome, the microbiota. It really paints a very complex picture. In essence, it's hard to get a handle on what's driving the biology. There's a need for a functional interpretation. And that need for this kind of functional handle steers directly into the value of metabolomics and some of the stuff Dave opened up with telling you about in this webinar. Because metabolomics produces a functional readout of the phenotypic state. Really, it's a surrogate of the phenotypic state. It's a readout that also aggregates all these influences, the genetic influences, the environmental influences, the microbiota influences. And because of this, it provides a consensus report on those influences and how they affect the phenotype, a consensus report and the associated biomarkers. And what's important about that is that the information embedded in that consensus report and those biomarkers are also translatable across species. So a couple of things that, just to elaborate on that point, we've had a lot of work with sponsors the last several years that have really illuminated that in terms of the genetic influence side, metabolomics is essentially a genome sentinel. That is, it can actually mark active genes and also help define their function. And on the right side, the external factor side, I'm using the microbiome as an example here. The work we've done with our sponsors has also shown that it can really illuminate the functional connection between the microbiota and the host phenotype. And these are all referenced at the end, as Dave mentioned. We're not going to go into details here, but suffice it to say, it elaborates on the point that the metabolome is providing this functional connection to the phenotype. So the last major driver of why we're seeing a shift to the clinic and translational studies is the fact that we have advanced metabolomic technology. This graph here is just showing our particular platform. And the orange trend line is showing that what we're able to do is cover more metabolites, more of the metabolome per sample across time. And at this point, we're looking at around 2,000 metabolites in a single sample. So in essence, your ability to discover sensitive and specific biomarkers has improved dramatically. The blue trend line is really important, particularly from a clinical perspective, because we've also driven down the process variability dramatically. And because of that, that's really enabled and opened up a lot of clinical opportunities. So just to elaborate on some of those things that this advanced technology has enabled, on the left, as I already kind of mentioned in a previous slide, it's enabling genome discovery and large cohort phenotyping. In essence, it's illuminating new target opportunities and biomarkers embedded in the genome. On the right, it's enabled individual disease and health assessment in the context of inborn errors as well as relatively healthy states. This has really kind of shown that at the individual level, metabolomics is opening up really a new frontier in precision medicine. And one point that on the bottom here that I didn't have a chance to dive into in the previous two slides, but is really a critical point. It's been discovered that an individual's metabolic profile is actually well conserved or stable across time. So unless someone is disrupted in terms of their health status or disease, the metabotype or their metabolic profile is very stable. So it makes it very tractable in terms of tracking individual health and response. So those are the four major drivers. The next few slides, we're going to talk about how people are now applying it in their project work. And I'm going to let Dave take you through and introduce that topic in the next slide.
So, in the preceding slides, we've covered metabolomics as an analysis platform designed to produce a phenotypic profile that reflects the metabolic status of the body. Based on the extensive work and studies conducted to date, it is not surprising to learn that some groups are now leveraging the platform to address questions that arise in moving from discovery and preclinical phases to clinical development. In the next sequence of slides, Kirk is going to cover examples of how the platform is being used to better define models, patient profiles, safety and efficacy readouts, and trial design. One observation at this point, though: to make the most effective use of the technology, teams should discuss the role of metabolomics as early as possible in their project planning.
Okay, thanks, Dave. So one of the first applications really is looking at models of disease and trying to define translatable biomarkers and also what the best model might be. The idea there is that it can really enable the target validation and also the pharmacometric modeling stages of R&D. And in this example, the investigators were looking at atherosclerosis with a statin treatment, and they looked at 24 different models, and they were essentially looking for what are the models that most closely replicate the human case at both baseline and then treatment. And also, what were those biomarkers or signatures that best reflected this? What this graph shows, the results of that study, if you just look at the graph and focus on the bottom with the dyslipidemic human, is you see that the number of models are either more closely matched with these various statistical tests or more distally matched to the human case. And surprisingly, a number of the more commonly used models were actually quite distant relative to a number of other models. And effectively, this allowed them to select the right model for the right target. But also it allowed them to carry these biomarkers forward to help inform in their drug development program. So once translational biomarkers are identified, you can now use the markers to help maximize the information that you glean from a clinical trial as well as the subsequent trial design. Because often these clinical trials are limited by a lack of biomarkers that are tightly coupled to target engagement and efficacy. And this can be particularly true in cases for novel first-in-class mechanisms. Often these trials rely on a single endpoint or a limited set of markers. So the idea with metabolomics is you can get an enriched panel of biomarkers. In essence, so you can more effectively model the PK-PD relationship for both desired and undesired effects. And, really the take-home is that good data, rich data, can help you generate rich, high-quality models as well. So in this example, this is a phase 1 biomarker-driven modeling for a novel first-in-class mechanism. And this really reflects a lot of the activity that we see our sponsors using the technology for today. In this example, they were using metabolite biomarkers that they discovered in those translational studies like I described in the previous slides. They were using them to define both efficacy and target engagement. And essentially they were using them in a phase 1 dose escalation study to look at PK and PD as well as efficacy. And using this more biomarker-rich data, they were hoping to model much more effectively and design the phase 2 study with even greater clarity. And really the take-home from those previous few slides is the clinical implication here is that robust, and as Dave mentioned, very early, doing this very early in preclinical development, robust translational biomarker discovery can deliver a nice suite, an enriched suite of biomarkers that you can model PK, PD, and efficacy to ultimately provide a deeper line of sight into the subsequent design and then ultimately maximize the probability of success. So oftentimes sponsors are looking beyond just efficacy and PD. In this particular example, looking at a promising oncology target, fatty acid synthase, or FASN, the sponsor was looking for markers that could inform on which tumors would be more sensitive, or in essence, which subjects are more likely to respond. And the approach they took was actually with cell lines. They actually looked at it in vitro setting to look at which cell lines were most sensitive to their inhibitor. And what they found, if you look on the graph on the right and kind of the trend arrow across that graph, is they found a number of metabolite biomarkers that correlated really nicely with sensitivity to their inhibitor. And what was really interesting about this is, although they looked at mRNA and protein expression levels, they were not able to find correlates to the sensitivity. So this is an example where metabolites, that functional state that Dave talked about earlier, were able to deliver biomarkers that correlated with this sensitivity. So really the clinical implication from this example is that if your success hinges on selecting patients that are most likely to respond, having that, again, early preclinical biomarker strategy, including metabolites, not just genes and proteins, can enable selection. And the second point is a bit broader, but it really is that it echoes a lot of what's been published recently in a number of different examples, that the analysis of a tumor's metabotype or metabolic profile can really enrich the understanding of response and prognosis. So another application really is to use the technology to discover biomarkers that can identify at-risk subjects. So in other words, finding markers that can help to more precisely define who's going to be enrolled in a trial, or also monitor those. And this is an example from our own work at Metabolon in the context of insulin resistance. So insulin resistance is a state that can occur up to 10 years before the onset of diabetes. And the problem is the current glycemic tests do not measure insulin resistance. By the time those tests flag an individual as being at risk, they're really essentially emerging or in the diabetic state. So we sought to define a test that could measure and identify subjects who were insulin resistant. And at a really high level, what we did is we started off, if you look on the left, with a very broad metabolic screen looking at about 500 biomarkers in a discovery cohort. Those top hits, those metabolites that correlated most with insulin resistance, were then taken into targeted assays in an expanded cohort. Then a validation cohort, along with final algorithm development, ultimately revealed an algorithm or test with 4 variables, including 3 metabolites, that could then identify subjects who were at risk for insulin resistance. And both of those results were published and referenced below here. But I want to just show you one way that the test has been used that's actually quite useful from a clinical development perspective. So the test was actually used to retrospectively analyze the samples from a clinical trial. This was a trial looking at pioglitazone and its ability to prevent the onset of diabetes in pre-diabetic subjects. And if you look at the plot on the bottom, what you can see if you just look at the pioglitazone group and the HbA1c, which is a established diabetes marker, you can't really see any difference between the red and the blue box. There's really no change. In contrast, if you look at the plot on the right, labeled Quantos-IR, which is the name for the test, you see a really nice response with the subjects at the follow-up time point, meaning that their insulin sensitivity had improved. And this was all published in the Journal of Clinical Endocrinology last year. But really, it just highlights the potential utility of finding these early sensitive biomarkers to stratify and identify subjects at risk, but also monitor treatment. Just as one other example of doing essentially the same thing that was done in the insulin resistance example, this is actually kidney disease, end-stage renal disease. And the idea here is there's a need to find biomarkers that can identify subjects very early on, which would potentially, allow one to enroll those subjects in an appropriate trial much more precisely, but then B, also be able to monitor efficacy with new agents targeting earlier aspects of the disease. So in essence, they looked at baseline biomarkers of a cohort of subjects with type 2 diabetes, and then they followed them for 8 to 12 years to find out which ones would progress to ESRD. And they wanted to explore which baseline biomarkers could predict that. They were able to find a lot of biomarkers shown here in this plot that associated with the outcome very early on. So this is 8 to 10 years before the progression of the disease. And this was published a couple of years ago in Kidney International. Most recently, there were 2 publications that showed really robust replication of the top biomarkers from that study in 3 additional cohorts represented by these 2 studies. So this really underscored, I think, really how metabolites can be really sensitive biomarkers or leading indicators of changes in physiological states. And really, I think both of those examples, if you consider the clinical implication, they show that metabolite markers can define early disease or risk potential, which ultimately can inform more precise and efficient trial design. One last thing we want to touch on here is the concept of using the technology for post-market differentiation. Since metabolites, as we've shown on a number of examples today, are actually sensitive to phenotypic or physiological changes, using the technology to detect those changes can provide a clear way to demonstrate favorable profiles of a drug. So we're seeing a lot of sponsors that are now actually using this probe of the physiological state, metabolomics, to actually differentiate the effects of a competitor molecule versus theirs. So a few examples to note on this slide, and again, it's not limited to what I'm putting here, but these are just obvious examples where we're seeing sponsors start to think about using the technology in post-market examples. Would be any case where an agent might induce weight gain or weight loss, where you would see obviously changes in lipids and ketone bodies, metabolic syndrome in the context of certain antivirals, or even any kind of GI side effects where you might anticipate the microbiota could be involved. So in the context of the post-marketing studies, the clinical implication here is that using metabolomics at that stage can reveal those subtle underlying physiological differences that might not be apparent by other methods that are essentially less sensitive to phenotypic changes. And therefore, ultimately, they can aid in differentiating the drug from competitor molecules. So at this juncture, I'm going to turn it back to Dave to just reflect on the technology for a little bit after we've wrapped up this section on how people are applying the technology.
So a quick recap, Kirk. We've covered a lot of ground talking about metabolomics and the applications at various stages of discovery and development. In the next few slides, you're going to highlight what I believe is the most important component of Metabolon's platform, and that really is the bioinformatics capabilities that enable the transformation of the metabolite profile data into actionable knowledge.
Thanks, Dave. And just on the next couple of slides, I'm going to show how you can generate data and ultimately take it to knowledge with the platform. And really, at a high level, this particular slide shows how, in essence, you can take any sample type and screen the entire metabolome to reveal those key biomarkers and insights. So the idea is, in this stage, taking a sample, generating data, and ultimately some form of information. But the information to knowledge piece really comes in the bioinformatics. So these are this slide shows a series of tools, in-house tools we've developed that we use to analyze the data that many of our sponsors also have access to. And essentially, these bioinformatic tools are critical for triaging this complex data down into just a few focused ideas that can then be further explored. So the bioinformatics is a critical piece, but to wrap it up, the scientific review process of actually having PhD biologists and biochemists interpret the data and review the data, that's where you establish a deeper level of understanding and ultimately create value. And that is ultimately packaged up, as I've shown here on this slide, into a final report where all that information is synthesized in a very intuitive way and then supported by pathway enrichment, pathway analysis, heat maps and plots, and then ultimately, the data file that can be piped into other informatic tools as well. So I'm going to go ahead and turn it back to Dave to offer a few closing comments. Hopefully that gave you a clear picture of essentially the process of taking data into knowledge. I'm going to let Dave wrap it up with a few closing comments.
Thanks, Kirk. I'm going to go through a couple of the key points that we've covered in today's webinar. Metabolomics is clearly a value-adding technology that has broad utility across the target to clinic continuum. We've shown quite a few examples today, and references for those examples will be in the appendix for this presentation, which will be available online. Metabolomics brings the genome to life through the generation of a phenotypic surrogate, which is essentially a consensus report of the aggregate changes from complex human systems biology. Metabolomics will identify biomarkers that can be used to track and monitor changes in the body's metabolic status, thereby enabling a more accurate characterization of patient populations. This is an extremely important consideration in the context of trial design. Finally, metabolomic profiling can facilitate target identification and validation through the functional reduction of genomic data. And Kirk, I just want to make one more point on the value proposition here. This is a technology that helps you assess patient population variability. It allows you to measure the responses of that heterogeneous population at the individual level in terms of baseline measurement and then following treatment with a therapeutic agent. It also provides an interpretation of that data in the context of substantial, validated biochemical knowledge. I could see how this technology could be of enormous benefit in understanding the mechanism of action of a therapeutic agent. That knowledge would inform a better decision-making process about options and risk for that molecule. Some closing comments from today. Metabolon's platform represents leading technology in the metabolomics arena, delivering data with breadth, accuracy, and precision. The sample-to-solution process not only involves state-of-the-art analysis technology, it also means that each project is partnered with a team of PhD-level scientists, a statistician, and study director. That team not only guides study design and execution, it also delivers comprehensive reports that provide critical analysis and interpretive guidance. Finally, I just want to mention Metabolon's track record of accomplishments includes over 4,000 studies completed and greater than 500 publications in top-tier peer-reviewed journals, together with significant partnerships with leading investigators and institutions. Thank you for listening to today's webinar. Kirk is now going to run through a couple of announcements and obviously tell you more about the exciting opportunity to engage the platform with 2 study opportunities. Kirk?
So let me just wrap up with just a few announcements here. Thanks again for listening today. But if you do have any inquiries, scientific inquiries, or any questions, feel free to send me an email, follow up with me. I'll be very responsive and happy to answer your questions and extend the dialogue here. The other thing just to note, if you're interested in following up and want to learn more, is we have a couple upcoming events. Our speaker here, Dave Morris, as well as some of our scientific staff, will be on the road March 21st to 25th, visiting clients on the west coast of the U.S. Also, April 25th through 29th, visiting clients or potential sponsors on the East Coast of the U.S. And this would be a really nice opportunity to talk more about the ideas we presented here in a fairly compact fashion. So if you want to schedule a meeting to speak with Dave or some of our scientific staff, just send Karen an email. And then, as we pointed out in the beginning and Dave mentioned, we do have a pilot program opportunity where 2 of the listeners today can receive a free pilot project based on a research program that they have an interest in. After this webinar, you'll be sent an email with information about it and also information on where you can download an application for this. But essentially, trying to take some of these ideas from this webinar, think about how you might want to use the technology, and we'd really like to explore different ideas with you in terms of these pilot projects. So we do encourage you to download the application and send us your idea. And we did want to just mention, Dave mentioned it several times during the webinar, but references from all of the things we went through rather briskly today are included in this slide deck. And now we'd be happy to take any questions from you.
Thank you for your presentations, Kirk and Dave. Before we begin the Q&A, Kirk needs to make an announcement. Kirk.
Yeah, so it's just regarding the upcoming events. Since we sent you guys the slides, there's been a brief update to the West Coast Roadshow. Instead of the week of the March 21st to 25th, it'll be the next week, the 29th to April 1st. So that's it, Jay-Sean.
Excellent. Thanks, Kirk. So now it is time for the Q&A. To ask a question, all you need to do is press the Ask a Question button on your console, type your question in, and then press Send. So our first question, and it asks, what about the potential of metabolomics to help unravel orphan drug MOA? Kirk?
Yeah, so no, that's a really good question. Just in general, a lot of tools that are used for MOA, maybe many people are familiar with things like chemical proteomics. As Dave and I highlighted in the webinar, since metabolomics is diagnostic of the phenotypic state, it's really a great way to unravel the MOA of any compound. And I think one of the benefits is that the signatures that you might get from compound action are actually, because they're mapped to pathways, are a really good way to then track back to the primary target of a drug. And I don't know if you have anything else to add, Dave, but.
No, I think that's a great comment, Kirk. I think the one thing that, in working with a lot of omics technologies over the years, the one thing that was most appealing to me about metabolomics is the fact that you see this end result of all the aggregate changes, all the integrations, and it manifests as this, amazing phenotype, this biochemical phenotype, and it's the precision and accuracy with which the Metabolon delivers that data is really astounding. So, I'm a big believer in the metabolomic platform, and I think it would be able to answer a lot of questions, not only in the phases that we've described today in the seminar, but there are a lot of other applications that could be used in this setting.
Excellent. Thank you both. So our next question comes from Jagan, and he asks, he was basically wondering how you would validate multiple organism biomarkers. So in his example, he says, suppose there is a metabolite marker in worms infected with Candida. Can your technology find biomarkers on both the host and the pathogen?
Yeah, so I'll go ahead and take the first part of that, Dave, and chime in if you have anything else to add. So just in general, the idea of finding markers that would translate across species is I think, one of the strengths of metabolomics. We often talk about different models and validating them across different species as really, if we don't see that type of translation, that it often is due to the model. But the other part embedded in that question really is about trying to tease apart a pathogen-host relationship. And the reality is, very often we'll get signatures that are very specific to pathogens or very specific to host. in those type of situations, the best thing to do is to try and incorporate a kind of a multi-omic approach where you can look at essentially what are the pathogens present, and then what's the metabolic profile, and try and combine the data sets so you can identify kind of the culprit as well as the metabolic signatures and how they relate to affecting the host.
Excellent. Thanks, Kurt. Dave, is there anything you'd like to sort of add to that?
No, obviously, I think if you can get the profiles for plus minus the pathogen, that can also inform, the specific organism-related profiles. So I think the data that I've seen from studies that Kirk was describing suggests that it's actually quite possible to discriminate those two sets of profiles, though, in that relationship.
Brilliant. And our next question asks, what kind of biological samples are generally sort of preferred when analysing the metabolomic profile in patients?
Yeah, it really depends on the study question. Generally, the default would be plasma or serum, sometimes urine. It could be CSF or saliva. If it happens to be oncology, it could actually be biopsy sample. But it really just depends on the question. If someone wants to get a really deep mechanistic insight, the more proximal we can get to where the action is occurring, the better. But it really, again, study design dependent.
Excellent. Is there anything else you'd like to add, Dave?
No, I can't add anything more to that, Jason. That is a great answer to the question, and there's been many examples of the Metabolon's ability to do that.
Sure. And our next question asks, are there any challenges, limitations, or bottlenecks to measure, detect metabolites precisely and accurately?
Yeah, boy, I'll try and keep that brief because that could be a whole webinar series here. But yeah, there are a lot of challenges to doing that. And really the main thing, if you go and you survey all the KOLs in the field of metabolomics, the biggest thing is identifying the metabolites. And that's one thing that we've put an immense amount of effort on at Metabolon through building essentially a really vast chemical library, and then building software that can essentially take those ion features and annotate them to actual metabolites. So that's probably the biggest one. There are a lot of things underneath all of that, and there are a lot of other elements, but that's probably the biggest challenge.
Yeah, I could just add on top of that, Kirk. It's a great question, and I think one of the things that's really important to consider as you're contemplating involving a metabolomic type analysis to really plan early for what you're going to do with your data. Make sure that you engage, whoever it is, very early in the design of your experiment, so you know which samples you're going to collect, and you know how many of them, and you know what time frame you're working within. So, I think again, I made the point in the webinar today that I think the early planning is really key, and there are probably examples where, again, a little bit of time spent up front at the design of stage of the study is going to pay off downstream when you're looking at the data coming out of the studies.
Excellent. And our next question is from Nasir, and it's quite a sort of specific question, but he asks, can metabolomic be used to identify the causes of extreme sensitivity to light?
Well, actually, that is very specific. Gosh, I've been at Metabolon doing metabolomics for almost a decade, and I don't recall that type of study. But I guess what I would say is, just in general, if there is a physiological difference, hence sensitivity to light, or any kind of phenotypic difference, you have the ability, as I think Dave and I tried to convey here, to find a metabolic signature of that. So I would say, given the vast physiologies and phenotypes where we found signatures, I would say yes, it would be worth a try.
Excellent. Thanks, Kirk. And our next question asks, so the examples offered in your presentation were very well established, sort of more common diseases. Can you comment about the role of metabolomics in rare diseases?
I'm going to definitely let you add some color to this, Dave, but I'm going to jump in initially. So really the principles are the same as common diseases. It's just a way to understand the mechanistic drivers of the disease and find biomarkers. I think one of the really interesting opportunities with rare diseases, oftentimes there's a driver gene that's identified, but sometimes it's not necessarily druggable. By looking at metabolomics, you can find some potentially downstream effects of that could be potentially targeted and it could be more druggable. And there's actually some nice examples. Actually, the example that, it might not be a rare disease per se, but the example Dave mentioned in sickle cell disease is a really nice example of that, where we know what the defect is, but targeting it effectively, therapeutically, has been a challenge. That metabolomic study that Dave cited actually found a downstream pathway that shows some therapeutic potential.
Mm-hmm.
Dave, would you sort of like to add any sort of colour to that?
Yeah, the only thing I can add to that, Jason, is there's a lot of interest at the moment in finding tools that can profile rare disease patient populations. As Kirk mentioned, often the rare disease manifests in a certain way, and I think using this technology, you could definitely see, a biochemical fingerprint of what that disease looks like. And again, as Kirk mentioned, it might illuminate a better ultimate intervention point in the treatment of the disease. Many of these things are obviously monogenic disorders, but they have consequences metabolically downstream, and it may be that one of those downstream checkpoints represents, as I said, a better intervention point for the treatment of the disease.
Thank you both. So just a quick reminder to our audience, we still have time for some more questions. Remember, to ask a question, all you need to do is press the Ask a Question button on your console, type in your question, and then press Send. So our next question, and this one's from Isabella, and she asks, would it be feasible to study INPC's metabolome analysis using your platform?
Oh, yeah. So we do a lot of different cell culture work, from stem cells to a lot of work in immunometabolism these days as well. So yeah, certainly. I mean, again, it depends on what's the specific question, designing a study around it. The samples are certainly feasible on the platform, and we've run those type of samples before. But it really often will come down to sample quantity. So.
Excellent. Thanks, Kirk. And our next question is from Richard, and it asks, is it necessary to sort of standardize sort of diet or fasting status when you're sort of carrying out the sort of metabolome analysis?
Yeah, so no, that's a great question. And the one thing I always say about that, because it's a very common question, is that just like with almost any other method, that's a best practice. The more variables you can reduce, including diet, time of day, things like that, the better off you'll be. It's not absolutely necessary, but it is definitely a way to make the study sharper, the results cleaner, the analysis cleaner.
Excellent. Thanks, Kirk. And this one's another question for you, and this one's sort of regards to sort of clarifying one of your sort of slides, and this was to do with sort of slide 33, 34, and they were wondering if you can sort of specify which pathway database you have been using.
Oh yeah, so all of our analysis tools are in-house tools we've developed, and there certainly are options out there outside of what we use, but the reality is we've looked at so many data sets, we've just very precisely and carefully annotated the pathways. So we basically plug right into our own bioinformatics workflow and tools for our analysis. Now, the nice thing is a lot of those tools are actually accessible to our sponsors as well. So when they get their results, they get their data set, they can actually access and play around with the tools as well.
Brilliant. Thanks, Kurt. And our next question asks, how well do your findings translate across species?
Okay, yeah, I tried to touch on that in one of the other answers, but the reality is this is what's really interesting. I think that with this technology, because it is so close to the phenotype, pathways are so well conserved, that it sounds like a bit of a cop-out, but the reality is if they don't translate, it's often because the model's just not that faithful. The reality, and the other point I'd like to make around that is we rarely will take a single metabolite or biomarker and try and hope that a single signature or biomarker will translate. Usually it's a collection of metabolites or pathways or themes that come together, let's say in an animal model that suggests a particular theme is important to, let's say, drug action or response to the drug. And we're carrying that whole theme forward into other animal models in humans. So in terms of being able to see those themes replicate, we've seen that time and time again. Again, if it doesn't translate, it's usually because the model's not that faithful.
Excellent. Thanks, Kirk. Dave, is there anything you'd like to add to that?
No, I think Kirk's highlighted the challenge there really well, Jason. I know people go looking in models for the same markers, and the fact is that some of these models they're not manifesting in those particular animal models. And then I guess the inference is that obviously anything they find, is it predictive of the human? There are some elements of the models that are translatable, but they definitely, I would not go as far as saying that they are predictive. But again, it depends on the model that you're dealing with and how it translates into the human situation.
Excellent. And our next question asks, how well do the results replicate?
Oh, yeah. Kind of along the same lines as the translation. If the study is designed well and you use a really good technology, they replicate really well. And there's a lot of examples of this. If, for example, you're, again, just using maybe a technology where you're only panning a few biomarkers and you're just wanting to take, let's say, citrate through, rather than a whole biological theme, I think you're tending to play with fire there. The signatures we find are not just a single metabolite. And because of that, and because we're usually carrying pathways or themes through, there's a really good precedent for them replicating.
Excellent. Thanks, Kirk. Sorry, Dave, was there anything you'd like to add?
Yeah, I'd like to just add one last point to that. Really, I think if I had to pin replication on anything, I would say the most important thing is the design of the study. It's not really the technology; it's the design of the study.
Excellent. Thanks, Kurt. Sorry about that. And our next question asks, and this one's from Arun, and asks, do we have enough information now for the use of the technology in the more effective use of drugs in clinical sort of setup? So actually, I'm gonna let Dave comment on this, because I've been working with Dave quite a bit the last several months, and one of the things he seems to really be enthusiastic about is the whole idea of, I think we touched on it in the webinar, getting kind of a richer set of biomarkers for modeling. So I'll let you elaborate.
Yeah, it's a great question, and actually I think it was probably, as you probably sensed in the webinar today, it was one of the messages that we were hoping to land with the audience, was that as much as we've done in the sort of preclinical early discovery space with this platform, it offers tremendous potential in clinical development, and largely through the ability to identify all the potential things that are changing plus minus treatment. Now, I think, as we mentioned, the ability of the platform to resolve a very large number of metabolites is certainly there. Once you've done that sentinel scan across the metabolite profiles in your patient populations, you can actually identify most likely subsets of markers that will allow you to track probably issues or markers related to efficacy and then potential liabilities. The other thing I think we tried to really emphasize today was that if you have a richer set of markers, it's a case of if you're always going to keep looking under the lamppost, you'll find the same things. This technology allows you to identify the things that may be changing in greater magnitude than the ones that are the most obvious. So you will be able to really track and monitor disease progression and plus minus treatment in a much more accurate and detailed way. It will also give you the opportunity to stratify your starting patient population and really understand their individual metabolite profiles and monitor those changes as you go through the trial as well. I think it has tremendous promise as a tool to be used even in phase 1 trials, and it would enable much more detailed pharmacometric modeling, and that then would inform probably a much more efficient trial design, because you would be able to look at a richer data set per patient and take that richer data set with you as they go through their clinical trial. So I'm a very big proponent in the use of this technology in a clinical setting, and absolutely I think it's a technology that will become known more for that space than probably it has been already in the early discovery and preclinical spaces.
Excellent. Thanks, Dave. Kirk, is there anything you'd like to add to that?
No.
Excellent. Then we'll move on to the next question, which asks, what percentage of your studies are with academics versus industry? And is this being adapted sort of well with technology and pharma industries?
Yeah, I don't have the data right in front of me, but it's probably a pretty even split. Yeah, I would say it's a pretty even split. I'd say that the pharma side, it's really starting to maybe get a little bit more traction than maybe in years past, and a lot of it is because of a lot of the work we've done with academic labs and some of in essence leading the charge in a lot of different areas and showing where this technology can be used, as you might imagine. Some of the examples of this where we've had a lot of heavy academic use and now we're seeing a lot more interest on the pharma side are areas like microbiome research, immunometabolism, which touches on, of course, cancer immunotherapy, but also areas like autoimmune disease. And then I also think, we touched on it in the webinar, but just the large cohort phenotyping, genotyping, with the idea that maybe if we do enough people and we do it well, at least in some instances, we can potentially circumvent animals for validating targets.
Excellent. Thanks, Kirk. And our next question asks, can you comment on the sort of potential of metabolomics in the standardization of anti-malarial sort of herbal medicine, so the replication, PK, PD, and sort of clinical trials?
I'm not really sure how to tackle that one. Dave, maybe just the whole idea of PK/PD with any medicine in trials. Maybe you can comment on.
I'm pretty sure that regardless of the medicine, that the platform will be able to pick up the changes. We'll be able to monitor when drugs on board, and anything related to PK, pharmacokinetics. It will be able to resolve, and there are probably examples of where metabolomics have been used exactly for anti-malarial medicines. So my simple but somewhat uninformed answer, because I'm just not that familiar with the sort of area of malaria, is probably yes. It can certainly be resolved at the level of a metabolomic profile.
Excellent. Thank you both. And our next question asks, how well does the platform integrate with proteomics arrays or sequence data?
Oh yeah, that's actually probably that's really one of the staple uses of the technology. People have sitting on a lot of proteomic or expression data, and just trying to get a handle on what are the signals in that data set. And I think we tried to elaborate on the webinar with metabolomics being, in essence, kind of the end product of that and residing as kind of a surrogate to the phenotype. Metabolomics has been a really valuable tool in triaging those types of data and adding value to that type of data. And I would say the biggest take-home is that a lot of the sponsors we've worked with who use this the most and also combine it the most with RNA data or proteomics data, they actually tend to refer to metabolomics as being kind of the noise reduction tool, or they use it as kind of a bait to find out where the signature is in the data, and then they look into, let's say, the expression data or the proteomics data. So that's a very common staple application.
Yeah. Just to add on to that, Jason, I think, knowing what some of the work that's being done at Metabolon at the moment in the academic environment, we're aware that a lot of groups are sitting on a lot of data, genomics data generated, through the various platforms. And I think they're struggling a little bit to know what the next stages are to bring that data to life, as we said in the webinar. And I think Metabolomics is certainly a tool that could help them do that. But again, I go back to the point I made early on in the webinar. I think it's really important in when you're designing these studies to really think through how you're going to determine the functional consequences of the genomics-related changes that you're going to ultimately find. And I would just think through that whole scenario before you commit to just one platform without thinking through what the functional consequences are of the data that you're going to generate. I think this particular platform that we've talked about today provides a very significant additional level of contextual information that could help address some of the questions that initially probably were at the start of the genomics-related studies.
Excellent, thank you both. And the next question that we have asks, how do you think your sort of platform can help in the design of better clinical designs where the changes are seen after a very long period of time, or even sort of many years?
Oh, okay, yeah. And I'll let you elaborate as well, Dave, but I think just if we think about any of these really long developing chronic diseases, I mean, Alzheimer's comes to mind as well. I think one of the bigger opportunities, and unfortunately, it's not necessarily something that can be done quickly, but is to actually do kind of baseline biomarker profiling and looking at outcomes. So this, that takes a long time. But in the case of Alzheimer's, for example, wouldn't it be awesome and powerful to have very early biomarkers or signatures of disease? I mean, that's something that people are looking at right now. The other obvious thing, of course, is just potentially with any trial is just being able to have another potential readout of hitting the target or of efficacy. But in this case, efficacy might be a little bit more of a subtle endpoint, right?
Yeah, I think the key piece here is that, as Kirk said, I think having those baseline reads from your patient population is going to be a great reference point as those patients progress through specific arms of the trial. Obviously, there are certain drugs out there that had some issues well into their clinical life, and begs the question of whether we would have been able to pick up some of the sort of harbingers of undesired activities earlier on if we'd had this technology used at the very early stage of those trials.
Excellent, thank you both. And I think we've got time for one last question. And this asks, can you offer any additional detail on the project opportunity you mentioned at the end of your presentation?
Not an immense amount of detail. We really want it to be a kind of a blank slate for you to think about any ideas that come to mind after this discussion, this webinar. I think we are the studies are 50 sample studies, but really I think the important thing is to send us your idea. And then upon award of that idea, we'll get on the phone with you. We'll assign a study director who can work on the specifics of the study design with you. After you execute the production of the samples or pull them from your bank in some cases, We'll then run it across our platform, do all the data collection in QC. The statistics will be run. A study director will analyze and interpret the data, get on the phone with you, go over the results, and then work on bundling it up into a final report. So the biggest thing is just get your idea on paper, and then we'll upon award, we'll go from there.
Excellent. Well, it sounds like a fascinating opportunity for someone. I'm afraid that's all we have time for today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. I would like to thank Dave Morris and Kirk Beebe for their presentations and for answering your questions today. I would also like to thank the webcast sponsors, Metabolon, and of course you, the audience, for taking the time to be with us today. Thanks for watching, and I hope you'll join us again for our next webcast.